viewers. Today we are going to discuss the topic Fermentation Types 2, which includes submerged, solid state fermentation and surface fermentation. Basically, fermentation technologies can be broadly classified into liquid fermentation and solid state fermentation based on the physical nature of the medium in which the fermentation is brought about. If the medium is liquid, we can then further classify it as being surface fermentation or submerged fermentation. This means if we take a thin level of liquid and allow no agitation, then that is called a surface fermentation. While if we take huge quantities in a tank and allow agitation, then that is submerged fermentation, meaning the medium component cells and others are all submerged in the whole of the liquid medium. While on the other hand, typically talk about solid state fermentation, which occurs in matrices which are solid in nature. Further, the submerged fermentation, which is the most important and widely used type of fermentation technologies, can be divided into containing either free cell or immobilized cell. Now, after a classification scheme, let us see all of these individually. What do they mean? We have a pictorial representation of the several types we just discussed, as these showing you a bioreactor which is solid state. Then we have the submerged bioreactor here, and we can also see liquid state bioreactor typically in the lab scale. We will begin our discussion in detail with liquid submerged fermenter. A typical bioreactor like the stirred tank bioreactor can be a very good example of a liquid submerged fermenter. Here we can see a container full of liquid, 1/3 being left free for foam formation and other advantages, while the whole of the liquid can be mixed with certain structures called as the impellers. These are meant for mixing the contents within the bioreactor, which includes the cell, that is the culture, broth, cells or the inoculum, as well as any other components that can be incorporated into the system. Another type of bioreactor, which is also a very good example of submerged bioreactor, is a bubble column fermenter. The only difference with the stirred tank bioreactor, which we earlier discussed, is that there are no impellers. But the question is, without impellers, how can so much huge quantity of media get mixed? Otherwise, the cells will be in one place, the nutrients will be in another place, and there will be no uniformity of the product formation. And that is the reason here we use a bubble column, meaning the air that is introduced through the bottom through a sparger will be allowed to move in a direction, unidirectional fashion, so that the air itself brings about the mixing. That is what we can see here. The air that is getting introduced is doing all the mixing of the liquid, cells and other components which are present. Next in line is the airlift bioreactor or airlift fermenter. This is also one of the typical examples including in the submerged fermenter. Now what does it do? Here this is very similar to the bubble column reactor. The difference is we have a draft tube which may be internally placed. At times the draft tube may be externally placed. What happens is the sparger, which is meant for air introduction, allows the entry of air in a definite direction. The air rises up, allowing the mixing of the liquid and other contents as it moves up, and then the air falls down. There is density difference, and that is why the air will move up but the liquid will fall down, and that is how the mixing is brought about. We can use two different spargers in the two different sides so that the air will rise from the two different sides and enter through the center. So the draft tube is very important in allowing a typical direction for the movement of the air, which causes the mixing. So these are the three examples which are frequently used for the submerged fermentation. We discussed that submerged fermentation can be including free cell type or maybe including immobilized cell types. So here whatever we discussed just now was showing you the example of a cell-free culture in the submerged mode. So this is what we have come across. Now let us see what is this immobilization and what are these immobilized enzymes or cells used in bioreactors. Immobilization means making it immobile or immovable. It will not be moving. Now what happens is cells or enzymes often we use in industries. If we want to recycle it, it is difficult to purify, separate. And so if we can make certain systems inside the bioreactor which will hold these enzymes or cells intact and allow only the passage of the broth after the product has formed, then we can retain them and reuse them. That is why we have used these cells in the immobilized conditions, and the bioreactor needs to be modified in that extent. Here we can see immobilized enzymes. So these are small beads which contains the enzymes within them, and once the substrate is added, product removed, the enzyme system in the immobilized cases will remain within the reactor. You need not take them out, and that is why circulation, recirculation can be possible. We have another example here which is showing again the beads containing the cells, and the whole of the production can take place once or even more than once. Next, we are moving to see the solid state fermentation and thus the solid state fermenter. Now, these basically are slightly different from the earlier one. The first difference is liquid is used to a very minimum extent, and we call it nutrient in the form of water limited condition. So the matrix are made up principally of solid substances, and the fermenter looks somewhat like this. We have trays or racks into which the medium, which is typically solid in nature, will be entrusted, and they will be inserted into the bioreactor chamber. The bioreactor chamber is actually meant for providing the controlled aeration, pH, temperature and other requirements. This is also a typical example of solid state fermenter, which is tray in nature, and the cross-sectional view is shown here. We'll have several layers of trays, each containing, or rather each layer is an example of a bioreactor. Now, for understanding solid state fermentation, we'll first see what are the substrates that we are using. Meaning the matrix will be made up of certain materials. At times the material can be inert, or at times the material can be nutrient in nature. Most often we would like to use certain material which are nutrients providing as well, like cereal grains, or it can be simply wood chips here, or can be bagasse, that is the leftover material after the sugarcane juice has been extracted from sugar. It can also be paper pulp and many others, and these are nutrients in the sense that the material is coarse, it will provide the packaging properly, but also additionally has either some carbohydrate source or some protein source along with it. On the other hand, we can also use inert materials, like here we have a typical diagram of a vermiculite. But if we are using inert material, they will not be able to supply nutrients, and as such, they should be impregnated with a liquid nutrient broth. Water is essential, but in solid state fermentation, we use limited water condition. This is another example where we are seeing the solid state fermentation in a koji type processes. Solid state fermentation dates back to earlier days, while in the Asian countries people were preparing koji and other koji related products. Those were principally made in trays. We can see the bioreactor housed with several trays here, and then there are other allowances, like for example there should be a UV so that the internal bioreactor can be sterilized. There are others like the air blowers in the several position. Air inlet and air outlet are important. This is the inlet and this is the outlet. And then there is another one which is an humidifier, maintaining the exact moisture content within the bioreactor. This is another example which mimics, or you can say reminds us of the production of vinegar in the solid state fermentation process. This is a closed cabinet, while the packing material can be incorporated with the inoculum, which may be some mainly fungi, or it can be bacterium also, and then the nutrient solution is dripped in so that the product can be produced. Now, most importantly, solid state fermentation utilizes fungal growth. The reason is the mycelium has the ability to take up the nutrients or absorb it from the material, and they can grow in numbers. Aeration is also employed, so being aerobic, they can happily grow there and produce the product that we want to derive from them. Here we can see typical example of the solid state bioreactors, which you can see here lined in trays. So actually speaking, the trays are the bioreactors which are containing the solid substrate, and then the fermentation is brought about in these kinds of flat trays. Now, if we look into a solid state fermenter plant, what happens is the raw materials, that is the bagasse or whatever we have chosen as the raw material, are taken. Then steam is allowed to be entered into the chamber, which will sterilize the material. Then inoculum is added, and there are several sensors attached like CO2, oxygen, temperature sensors. These will tell us how long the fermentation should be carried on, or if required the temperature could be reduced or increased. All that can be viewed, and we can have other outlets and inlets like the cooling and the heating unit, which will allow the proper maintenance of ambient temperature, pressure, pH requirements within the bioreactor chamber for a proper production stage. Now, there should be certain important usefulness why we are going to use the solid state fermentation. Now, that includes lower capital cost, very important. We will not have to invest much. Then there are lower waste output. This is also equally important, and obviously less water is used. Energy requirement are less because there is no need for mixing, very important. It is simple and obviously can be used even in the home processes, as well as it is being used from tradition. Then medium are also very simple. It's easy for aeration. It's very economical, and storage of the dried material becomes even more economical. So on the overall process, we see solid state fermentation is very economical if can be applied. What are the uses of the solid state fermentation? We'll see them used in several enzyme production. Now, the enzymes which are produced by solid state fermentation includes hemicellulases, cellulases and pectinases. And where do they find their use? Pectinases are most often found being used in the food industry for several food processing plants. In the cattle feed or the animal feed, they use hemicellulases and cellulases so that the feed can be properly digested initially, and it is easier for the cattle to consume. Again, we have the brewing and the distilling industry which uses hemicellulases. The baking industry again using hemicellulases and cellulases. And finally, to wind up the uses with the enzymes, we have the production of bioethanol, the need for today, which uses extensively hemicellulases and cellulases. And other uses of solid state fermentation includes traditional food production like the sake, a drink, koji, soy sauce fermentation, and not to forget the mushroom cultivation. All these are produced by solid state fermentation. And this is another example where we have already discussed earlier about the soy sauce fermentation. So like that, the surface fermentation again in a picture for you. This koji production, which includes the mold, that is Aspergillus oryzae, grown on rice or barley. This is differentiation chart to give you an idea which is better, submerged or solid state. But actually speaking, it depends on the organism we are using as well as the product. Anyway, microorganism substrate is static in case of solid state but is agitated in submerged, as we have discussed. Water usage limited and unlimited. Likewise, oxygen supply by diffusion in case of solid state but aeration for submerged. Then volumetric production by fermentation is smaller in case of solid state, while we get large amount of product in submerged processes. Liquid waste is less in solid state, more in submerged. Physical energy and human energy are required less in case of solid state or high in case of human energy, while capital investment for an industrial sense, if we see, is very low in case of solid state while quite high in the submerged. Now we will be looking into the liquid surface fermentation. What happens here is basically the medium is liquid in nature obviously, but taken in large surface area so that growth of aerobic microbes can take place on the surface of it. Now if we take an example in the lab, we can use a culture tube or a conical flask, but here if we see the oxygen transfer in this case is less, while in this case it is more. So we prefer larger surface area for liquid surface fermentation. That is obvious. We can see here also this particular flask will give a better result if we are looking for liquid surface fermentation and products obtained through them. Now, this is typically shown to tell us that the microbial organisms, principally of the fungal genera Aspergillus or similar organisms, will generally take their nutrient from the liquid medium which is lying just below their substrate growth. Now, this is the surface culture of Aspergillus, and oxygen diffusion is the optimum on the surface, and whatever nutrients they require in the form of salts and others, they take it through their substrate hypha from the bottom, which is liquid in nature. And we can very well see the depth or the height of the liquid taken is very, very less. So, surface area should be more and the depth should be less. That is the prerequisite requirement required for any liquid surface fermentation. We will see in case of wine production, if we allow more surface area, and mostly they are carried out in open tanks at times, they will give us very good product. In the case, that is, aeration will be good on the top when they are initially growing, while yeasts will be growing at the bottom definitely to give us the product. Mixing is not required and the liquid can be just left static in that sense. Now, in most cases, this liquid surface fermentation is meant for, again, fungal mycelial growth of the nature of Aspergillus, Rhizopas, at times giving us products like mycoproteins or the single cell proteins from other bacterial sources as well. So, these are the list of products which we can derive by the liquid surface fermentation. At present generation, we prefer to go for submerged fermentation rather than liquid surface fermentation. The disadvantage being the use of huge amount of surface area. Space is a constraint, and as such, the liquid surface is traditionally used, while the modern ways generally employs a submerged fermentation mode for the same kind of production. That gives us better product and faster yield, obviously, with the submerged production. So, we have seen surface, submerged as well as solid state fermentation. They will be used depending on the need and the organism and the product wherever to be applied.